So, good afternoon and welcome to our weekly meeting. This is Caterina Cremonesi from Prodotti Gianni. Please silence your microphone and close your camera. Today, the talk is fully dedicated to SARS Cov2, and we have the pleasure to host Alexander Bol as a speaker. Alexander is a medical doctor by training and a senior scientist at Genetex. His area of expertise is in the field of viruses, oncoviruses, and mammalian chromatin. The title of the presentation is SARS-CoV-2 and COVID-19, a world problem needing a world response. If you have any questions, please write them down in the chat in English. Thank you. Alex, it's your time. Thank you for attending.
Katerina, thank you very much for the invitation, and thank you for all the attendees today. I'll go ahead and get started right away. Katerina already gave the title of my talk. Let me just give you a broad overview of what we'll be talking about today. First, the origin of SARS-CoV. Where did it come from? Where is it thought to have originated from? The second is the global situation presently, as of numbers from yesterday. Three will be the biology of SARS-CoV-2. What do we know at this point based on our understanding of SARS-CoV, MERS-CoV, and other coronaviruses? Fourth will be the clinical aspects of COVID-19 disease itself. And the final sort of chapter will be about Genetex's SARS-CoV product catalog and other products related to coronavirus research. So I have to introduce, maybe start with a little bit of levity, sort of more directed at my own country in the United States. I don't know what it was like in Italy, but what we found was that as soon as the shutdown orders came, toilet paper became like gold, and couldn't find it on the shelves. The second one is a little bit more grim, the fact that we have such a huge defense arsenal, and yet we're completely unprepared for this virus, so that even our physicians and medical professionals couldn't get simple masks to protect themselves while taking care of patients. So the origin of SARS-CoV, COVID-19, it really is thought to, even though there's some controversy about this, may have started in December 2019 in the city of Wuhan, which is the capital of Hubei province, China. Please forgive my pronunciation. Patients began presenting to the hospital with severe pneumonia of unknown cause. Most of the patients seem to be linked to Huanan Wholesale Seafood Market, which also sells live animals. And by controversial, I mean there's some indication that perhaps the exposure may have happened earlier out in the countryside. At the time, a SARS surveillance system, which had been put in place after the last epidemic with SARS-CoV, respiratory samples from patients were sent to reference labs. By December 31st, 2019, the World Health Organization had been notified. By January 1st, the Huanan market had been closed. By January 7th, Chinese scientists had identified the coronavirus involved as the etiologic agent, with greater than 95% homology to a bat coronavirus and greater than 70% identity actually to SARS-CoV. Exponential case increase was probably driven by Chinese New Year travel and human-to-human transmission. By January 23rd, the entire city of Wuhan, which is an 11 million population, had been locked down. And then there was the cases then spread to the remainder of the province. Epidemic doubling at the time was about 1.8 days, and by then it had escaped out of China and had gotten into seeing exponential increases in Italy, South Korea, and Iran, which were hit very early in the pandemic. And by March 11th, the World Health Organization declared SARS-COVID-19 a pandemic. When one looks at the virus itself, and we'll talk about this a little bit more in depth, what one sees in a similarity plot between the SARS-CoV genome and its closest relative, you could say, which is a horseshoe bat coronavirus called RATG13, where it shows 96% sequence identity, is that there's a great deal of variability actually in the receptor binding motif, receptor binding domain associated in the spike protein, so the region that actually binds to the ACE2 inhibitor, or the ACE2 receptor, excuse me. Looking at further studies from that same study, this is by Cagliani et al. They were found 6 bona fide positive sites of positive selection in NSP1, ORF8, and N. But what's most interesting is the changes that had actually occurred in the receptor binding motif within the receptor binding domain in the S1 protein. And what seemed to come out of it was that SARS-CoV-2 probably came from a common ancestor, sort of upstream of both the rat and SARS-CoV-2, with changes that seem to be selected for within the receptor binding motif found in the SARS-CoV, and then in some pangolin viruses where it's very, very highly identical, even more so than in the rat, indicating there possibly could have been some sort of recombination event in a progenitor that had these changes in the receptor binding motif and the rat genome, or the rat coronavirus genome. As a source of review, what we know is that we've had 3 major lethal human infecting coronaviruses come out within the last 20 years. SARS-CoV, all 3 of them actually began with the natural route being in the bat, and then 3 intermediary species that seem to be for each specific coronavirus. For SARS, it seemed to be these civet cats. For MERS, camels, and it's thought to be for SARS-CoV-2 pangolins. When one looks at phylogenetic trees, what we see is that the SARS-CoV-2 genomes that have been examined are most closely related to the bat coronavirus, and then also to SARS, and more distantly related to MERS. Ciario, in their study, believe that the jump to humans from bats likely occurred late November to early December of 2019, although there's some evidence that it may have actually occurred even sooner, perhaps as far back as September of 2019. So most evidence, just to summarize this one part, most evidence suggests SARS-CoV-2 originated in Wuhan, China, associated with the Huanan wholesale seafood market in December 2019. Other theories include that it originated in rural farming regions, where bat guano is used for fertilizer, and perhaps in another interaction between bat and man. 2 phylogenetic analyses. One puts it as far back as late November when we jumped to humans. A second analysis went to mid-September. A third analysis supports limited positive selection and a possible recombination event in the receptor binding motif found in some pangolin viruses as a common ancestor of bat-CoV RATG13 SARS-CoV that was poised for human infection. And once again, the SARS-CoV was declared a pandemic by the World Health Organization on March 11th of this year. When we look at the global situation presently, and this is data upwards as most recently as yesterday, what we see is the vast majority of cases now are in the United States, but there's regions in Europe that are multiple countries in Europe that are still seeing a huge number of cases, and mainland China. And we'll see this more in detail as we move on through the presentation. If you look at the number of cases per capita, what you notice are countries like Spain and Italy were hit the hardest in terms of, you can tell by the color. United States has also been hit hard, Canada, But now the area of interest, of course, is Russia. We'll see that in a moment. If you look at how countries are doing now, individual countries, in terms of where new cases are increasing, what you see is the populous nations such as Russia, Brazil, Canada, India, Saudi Arabia, and Sweden, and a whole list of other countries are still seeing increases in cases. And now, if you're looking at cases, countries where cases are remaining about the same, we see the United States, Spain, and Italy, United Kingdom, and Turkey, and also mainland China, among other countries, have started to level off. Where the number of new cases are decreasing would include countries such as Germany, France, Iran. Iran, of course, was hit very early hard. Belgium, Switzerland, Portugal, etc. If you look at the case growth rate over time, what you see is, by and large, many of the countries that had already been involved are starting to see a tapering, if you will, of their of the new cases of the growth rate. The main exception to that, of course, is Russia. Now, this list actually goes on much longer, and this all the countries. This is all I could get on the slide, though. But it does show that in Russia, there's still an ongoing issue of increasing cases. In the United States, what you find is that the Northeast is particularly hard hit. States like New York, New Jersey, Massachusetts, in the Midwest, Michigan and Illinois, and along the West Coast in Washington State, in San Francisco and Los Angeles, and in California, and then Louisiana, all had major, undergoing major outbreaks. Happily, though, and with some hopeful good news, is that if you look at the new reported cases by day across the world, and also the reported deaths, you'll see clear plateauing that seems to have been occurring. So it does still seem to be a country-specific problem, whether cases are going up, staying the same, or going down. But by and large, overall, in the global situation, things seem to be leveling off. So to summarize this part of the talk, as of April 23rd, yesterday, this actually, I just looked, this actually number is now 2.7 million. People have been sickened by SARS-CoV on 6 continents and 177 countries, probably in excess now of 180,000 deaths. Many countries impacted early in the pandemic are seeing their numbers of new cases stay the same or decrease, though other populous nations are still seeing increases. In the United States, hard-hit states included New York, New Jersey, Michigan, California, and also Louisiana. Worldwide, the number of new daily cases appears to be plateauing, as is the number of daily deaths. But until testing becomes more widespread, and we'll talk about that, this still remains unclear. And number 5 is, I think, something that we can all relate to, is that the return to normalcy as we know it, and the long-term effects on countries and the world economy remains to be seen, but at this point appear to be quite dire. In discussing the biology of SARS-CoV, we can make some general statements and then talk about some details. So as you know, coronaviruses are envelope positive sense single-stranded RNA viruses, about 60 to 140 nanometers in diameter. They belong to the family Coronaviridae. There's 4 genera: alpha, beta, gamma, and delta. For the most part, gamma and delta infect primarily birds and some mammals. There are 7 coronaviruses that are known to infect humans. 4 of those in the alpha and beta genera most often cause mild respiratory disease, also some serious lower respiratory tract disease in children. SARS-CoV-2 and SARS-CoV, which are more closely related, belong to a subgenus, the sarbecovirus, whereas MERS-CoV is more distantly related. It's in a separate subgenus, it's Mirbecovirus. If one looks at the actual genome organizations, what we find out is that SARS-CoV is just over 29.9 kilobases in size. It shares about 79% sequence identity with SARS-CoV, but only 50% with MERS-CoV. Revisiting revisiting something that we had spoken about previously about these mutations. So to sort of focus on notable features of the SARS-CoV-2 genome, the spike protein has 6 mutations in the receptor binding domain that appear to optimize binding to the receptor known as ACE2, but by an unknown mechanism in terms of it's not computationally predicted, which argues against the notion that this virus was somehow designed or leaked out of a lab. In addition, there's an insertion here of 12 nucleotides at the junction of the S1 and S2 proteins coded by the full-length S gene, excuse me, that generate a polybasic RAR cleavage site, which means more effective furin cleavage, affecting infectivity and host range. If you'll notice, I know it's hard to see, right upstream there's a proline, and this proline has been shown in other coronaviruses to lead to O-linked glycan addition to the boxed serine and threonine residues. And people think, or researchers think, that this could generate a mucin-like domain that might be important for immunoinvasion. And the possible scenarios that still need to be worked out, whether the progenitor that gave rise to SARS-CoV-2 and the RATG13, whether that was that progenitor genome, which could be still cycling out there, underwent natural selection either prior to or after transfer to humans, because there does appear to be some element of selection, particularly in this receptor binding domain. Though, as I said earlier, it could have been some type of recombination event that also showed up in this pangolin coronavirus sequence. The SARS-CoV life cycle is summarized here. At this point, what we know is the SARS-CoV-2 genome appears to code for perhaps 29 open reading frames, with 16 non-structural proteins, 4 structural proteins, and at least 6 or 7 accessory proteins. I'll go through this very briefly. It's positive sense. So initially, after binding and uptake into the host cell, the genomic RNA, which is positive sense, is translated. You get expression of the non-structural proteins, which include all the replicative apparatus. And then through a series of events that is still not fully understood, there's negative strand RNAs generated, and then nested set messenger RNAs generated that lead to the production of the accessory proteins and the structural proteins. Then the structural proteins are inserted into the Golgi and rough ER into this complex, and new virus is constructed and released from the cell. If we were to look at this a little bit more in detail, we can focus on the actual receptor binding. So SARS-CoV binds to the ACE2 receptor through its S protein. The type 2 transmembrane serine protease, TMPRSS2, cleaves ACE2 and S, and S is activated, which leads to viral entry. So TMPRSS2 is a cofactor involved in binding of the virus to the host cell. Both SARS-CoV-2 and SARS-CoV bind ACE2, while MERS-CoV binds dipeptidase 4, DPP4. It is sort of a general theme that coronaviruses seem to like to bind to peptidases, but the enzymatic activity of peptidase apparently is not necessary for that binding. If we look at the S protein itself, it's a trimeric structure. The S1 basically forms it forms the top of this here in the purple with the orange receptor binding motif that engages ACE2. The S2 region forms a stalk, and so this is what it looks like overall. And then here is actually the SARS-CoV spike protein through its SARS-CoV spike receptor binding domain, actually engaging human ACE2. And so as a summary to this part, SARS-CoV-2 is one of 3 beta coronaviruses known to cause severe and potentially fatal disease in humans. Genome size is almost 30 kilobases. Genomically, it shares more sequence identity with SARS-CoV, about 79%, than with MERS-CoV, which is about 50. Key genomic features include point mutations in the receptor binding motif that optimize ACE binding, as well as introduction of a polybasic site in the form of 12 nucleotides that leads to effective furin cleavage between S1 and S2. This addition also puts a position with a leading proline that seems to give a signal for O-glycan addition that may be active in immunoevasion. In addition, there are mutations from one of the early slides in NSP1 and ORF8, but these remain to be evaluated in terms of their effect on the transmissibility or pathogenicity of the virus. The SARS-CoV-2 genome codes for at least 26 to 27 proteins, though perhaps 29 ORFs are present, and the S1 protein receptor binding motif interacts with ACE2, like SARS-CoV, and entry is aided by the serine protease TMPRSS2. Moving on to the clinical aspects of COVID-19, this chart is actually instrumental and helpful in just sort of reviewing some of the things we know as a basis for this part of the talk. As we were saying, SARS-CoV-2 and SARS-CoV are in the same clade, more distant from MERS-CoV, which is in clade 2. The animal reservoir is for all three is bats. And then we mentioned the difference in intermediate hosts, the idea that ACE2 is the receptor for both SARS-CoV-2 and SARS-CoV, whereas DPP4 for MERS-CoV. Now, these two columns are particularly interesting. If we look at the case fatality rates for these two, what you see is MERS-CoV, in the way it was, and the effect that it had was actually quite a bit more lethal. SARS-CoV was 9.5%, so not as much, but SARS-CoV-2 presently is 2.3% for the case fatality rate. And this might actually ultimately end up being lower once we know the total number of people infected. But what's interesting here is the value to take into consideration here is known as the R0, the basic reproduction number, which is a measure of transmissibility or contagiousness. And if the R0 is greater than 1, the outbreak is expected to continue, and the value can reflect the expected magnitude of the outbreak. So what you notice for MERS, it's 0.7. SARS-CoV-2, 1.7 to 1.9, and the R0 has been, in different sources that I've seen, sort of reliably kept or put at between 2 and 2.5. So it does seem a little bit more transmissible than the other two coronaviruses that have affected humans. By way of comparison, to put this in some context, the seasonal flu usually has an R0 of about 1.3. The 1918 flu pandemic had an R0 of about 1.8. And measles, which everyone knows is highly, highly contagious, has an R0 of about 12 to 18. What are we seeing on clinical with patients with severe disease are coming in? And granted, we have to keep in mind that roughly 80% of patients are asymptomatic or have very, very mild disease. It's the other maybe 20% who have more moderate disease or very severe disease. And what are we seeing in the patients with more severe disease and even moderate disease? Is fever in almost all patients. Usually cough and chest tightness, shortness of breath, body aches. Some interesting findings where people lose their sense of smell, and whether or not there's indicates some neural involvement, which has not been fully understood yet. But this does seem to be a reproducible finding. In patients with very severe disease, the median time from the symptom onset to the actual shortness of breath is 5 days. Hospitalization 7 days, and people go into an acute respiratory distress syndrome with severe disease within 8 days. For those patients who actually recover, they're usually in the hospital for about 10 days. In the one study that we're referring to here, the average age was about 49 years old. Males tend to have more severe disease than females. That has been sort of again, a consistent finding. For the patients who come into the hospital, almost a third of them end up in the intensive care unit, either with high flow nasal cannula oxygen or on a ventilator. And in some lab findings, what you see is a leukopenia and a lymphopenia, increased AST, ICU patients that are showing evidence of more of a higher inflammatory state show a higher PT and D dimers. The increased troponin might indicate a myocardial injury. But what also seems to be fairly consistent is that when people do CTs of the chest, about 98% of people have bilateral ground glass opacity, and we'll see a picture of this, subsegmental consolidation. In addition, if you measure inflammatory cytokines, these are elevated. And what we'll talk about one in particular briefly is IL-6. Complications are, I think, well known to this audience. As mentioned previously, you get ARDS, acute respiratory distress syndrome, which means basically failure for the lungs to be able to do oxygen and gas exchange. Secondary infection can happen in 10% of cases, as well as acute lung and renal injury, not to mention shock. Treatment at this point is largely supportive. Patients go into isolation, they're in a negative pressure room. The staff has to be outfitted in personal protective equipment. Diagnosis is usually made with RT-PCR based testing. Again, largely supportive care. Sometimes you treat with empiric antimicrobials, sometimes neuraminidase inhibitors. People have been treating, we'll talk about this in a moment, with certain medications that are already known and were used for other viruses, remdesivir, lopinavir, ritonavir, and others, and no indication at this point for the need of steroids. Transmission, as we know, is primarily in respiratory secretions, but there's also live virus found in stool samples. The virus appears to exist for some time on fomites, and at this point, the incubation time that we've seen is roughly about 5 to 14 days. Diagnostic testing has relied almost entirely on PCR-based methodologies. There are 3 companies that make decentralized point-of-care tests. The one that's gotten a lot of press is Abbott's ID Now COVID-19 test that gives positive result in 5 minutes and negative in 13 minutes. If we look at some of the at the case fatality rate and get an idea of which patients are at most risk, what we see is that when you start getting above 60 years old, the case fatality rate goes up dramatically. As mentioned earlier, most of the severe disease is in males, and having any sort of pre-existing chronic medical condition puts you at much greater risk for having a severe or even fatal outcome, including cardiovascular disease, diabetes, chronic respiratory disease like COPD, hypertension, cancer. All leads to higher case fatality rates. Of note, apparently smoking has been shown to actually induce expression of the ACE2 receptor. So it's another reason to avoid smoking. This is a very busy slide, but basically gives sort of an overview of the pathogenesis, which you see with COVID-19. As we mentioned, in most cases, people in 80% of cases have mild viremia. Patients are asymptomatic or have mild symptoms. What the rest is happening here sort of focuses on this central thing and this notion of cytokine storm, which is basically the immune system response to the virus. That cytokine storm can lead to ARDS and then subsequently death. But also the pro-inflammatory cytokine profile that comes up can actually lead to shock and tissue damage in other organs. And I think this is an area here that is going to be looked at more. And this is not entirely surprising because ACE2, the ACE2 receptor is widely expressed in various mucosa all throughout the body. A notion that we'll come back to in a minute is that what we've seen in some patients is that those with a high IgG response to the infection can actually do clinically worse. And the reason for that is because of antibody-dependent enhancement of the infection and uptake of the virus because of antibodies that are generated against the spike protein, which most of the immune response is directed against. To revisit that notion of lymphopenia and leukopenia, which we had talked about earlier in the context of the cytokine storm, what we find is SARS-CoV activates both innate and adaptive immune responses, and the lymphopenia is common in severe diseases. what happens is CD4, CD8 T cells, B cells, and natural killer cells, and the percentage of monocytes, eosinophils, and basophils are all decreased in severe disease. An increased number of neutrophils or an increased neutrophil to lymphocyte ratio is seen in severe disease and is prognostic of a worse outcome. You also see exhaustion markers on the cytotoxic lymphocytes like NKG2A. Now, the good news is that what happens is for survivors, these levels all renormalize and actually acquire antibodies to the virus. Feng et al. Actually had an interesting study that showed that SARS-CoV-2 could directly infect macrophages, as macrophages, unlike some of these other immune cells, does express ACE2. Macrophages then express IL-6 that can actually cause lymphocyte apoptosis. In addition to that, exposure to viral antigens can activate T cells and B cells and leads to upregulated fast-dependent activation-induced cell death. And that's sort of shown schematically here with moderate COVID-19. You do get increased cytokine levels, but there's not a complete loss of lymphocytes. Here in the issue, when the cytokine storm happens, however, you get fever, cough, fatigue, myalgia, dyspnea, ARDS, and respiratory failure. We can explore that in a little bit more detail here. So once again, we'll just look at the top part of this figure. SARS-CoV through ACE2 infects the lung epithelium, the two types of alveolar cells. And then we have sort of the multicellular response, T cells, polymorphic PMNs, and macrophages. When viral clearance occurs, you can see that T cells are not lost, T cells are not destroyed. The NK cells are still intact, B cells launch a humoral response, as opposed to the hyperinflammatory response characterized by an enrichment of neutrophils, loss of lymphocytes, and macrophages kicking out IL-6, which are driving the conditions that we talked about on the previous slide. So part of the theory, or part of the attempt now to block this cytokine storm, is looking at drugs, at monoclonal antibodies, one being tocilizumab, which is an IV drug, and seralutinib, which is a sub-Q drug, which are both IL-6 receptor blockers. Now, as I said, they're already used for cytokine release syndrome and in certain autoimmune diseases, including rheumatoid arthritis. And some results from some early studies have shown some fairly remarkable recoveries with tocilizumab, but we'll have to see with further studies whether that still is the case. I'd said that I would show you an example of a CT scan from a patient with severe disease, and here's sort of a classic picture of what you see. This is a CT scan of lungs, obviously. This would be the right side, and what you see, these are in the white with the white arrows, the ground glass bilateral opacities that we see in lobular and subsegmental distribution. What you also see is indicated by the black arrows is pleural thickening. And then what you see with the red arrow is something that's really sort of descriptive of what's going on in terms of the pathophysiology of this. This clearness here, this what looks like a little twig, is basically a bronchiole, and basically it's outlined by this radiodense material, which is essentially alveoli that are just filled with fluid. So they are incapable of any more gas exchange, and they are so packed with debris and inflammatory matter and fluid that they are incapable of functioning. So they perfectly outline the airway of the bronchiole. As I said, therapies are now moving in, and what people have tried to do in the use of time and to save lives, our studies are looking, first of all, including one by the World Health Organization, to see if they can repurpose drugs that are already in clinical use for other applications and therefore have acceptable toxicity profiles. And so we talked about a couple of them earlier. Remdesivir was actually designed as a RNA polymerase inhibitor. I think it was initially designed for Ebola. Chloroquine and hydroxychloroquine, which was championed by our president for some time, it functions to raise the pH in the endosome and hopefully break the cycle here. But studies on this have shown this not to be efficacious, at least in the early studies. The same thing with remdesivir. There's anecdotal success, but for the most part, early study that came back indicates that it doesn't particularly, it's not a game changer, so to speak. Same case with lopinavir and ritonavir, which are protease inhibitors, initially designed, I believe, for HIV. Camostat mesylate is actually used in Japan for treatment of chronic pancreatitis pain, and it's actually a serine protease inhibitor. So people are thinking that it might be useful to inhibit the activity of this cofactor involved in virus fusion, membrane fusion, and uptake. And as I mentioned, there were a couple of monoclonal antibodies, but there's also maybe treatment that would, or monoclonal antibodies directed against spike protein that may be useful. And then also what people are looking at is the use of convalescent plasma. So that means plasma from survivors of COVID-19 who may have antibodies that could be therapeutically used for other patients in severe cases. So the emphasis now, of course, after PCR testing, making diagnosis, as countries around the world start to see their COVID-19 case number plateau and then hopefully wane, what's important now is antibody testing. And the key is to determine who is immune. Particularly, this is particularly important as countries try to remobilize their economies and workforce, and of course, most important for the healthcare personnel. Many of these antibody tests have been going through expedited development with less stringent FDA assessment criteria in the United States, for example. This sort of testing will also be crucial, very crucial, for assessing vaccine development to evaluate effectiveness, and also contact tracing. It will also inform public policymakers about the true number of asymptomatic cases. In other words, people who didn't even know they were sick, have they still been able to generate? Are there people walking around who've had a mild disease or not even known they were affected, and yet still were able to generate an immune response? The time required to develop an effective antibody assay greatly exceeds a PCR test. And I heard one scientist say, basically, with a PCR test, the genome sequence today, you have the PCR test the next day. Of course, the primary target is spike protein, but also people are looking at nucleic capsid. And reports are there are at least 10 antibody-based tests being used in Chinese hospitals as right now. One key issue to detect, in the value of detecting antibodies to SARS-CoV is to make sure you're actually detecting antibodies that are specific to SARS-CoV-2, as opposed to other coronaviruses. So we want to make sure that you're not picking up antibodies that may not be as effective to, say, SARS-CoV's spike, which had 75% identity, or perhaps 50 to 60% identity with the other human coronaviruses that people might have been exposed to, but can cause much less severe disease. We mentioned those at the beginning. So the idea is to identify antibodies that are SARS-CoV-2 specific. And then, of course, the million-dollar question is immunity lifelong? Some experts think yes, maybe T-cell based. And this is based on follow-up studies done with SARS-CoV survivors. Now, these people are being like 17 years out. There's some optimism for this, but it still remains unclear. Vaccine development is presently being pursued by at least 40 pharmaceutical companies and academic institutions. And they're using different approaches. They're using whole cell killed or live attenuated virus vaccines. They're using recombinant protein subunit vaccines, sometimes very specific regions, say the receptor binding domain of spike. They're also using nucleic acid vaccines, or testing nucleic acid vaccines, or specifically messenger RNA. Efficacy is being assessed, hopefully using mouse models expressing the human ACE2, and also virus-like particles. And just as a reminder, remember, the vaccine makers have to be somewhat careful because of the antibody-dependent enhancement issues that came up when you use full-length spike as an immunogen. Has been shown in the past to cause severe liver damage. And in addition, there's also what's been noticed is immunopotentiation with eosinophilic infiltration or increased infectivity has also been seen with whole virus vaccines. So care has to be exercised as vaccine developers move forward. So the clinical aspects of COVID-19, to summarize this section, SARS-CoV-2 has a lower case fatality rate than SARS-CoV or MERS-CoV-2 or MERS-CoV, but appears to have a higher transmissibility based on that R0 number. Clinical progression in serious cases from onset of symptoms to hospitalization and in severe cases ventilatory support can occur within the span of 8 days. There does appear to be a greater male over female fatality, and the highest risk group are people over 60 years with pre-existing conditions. Pathogenesis involves cytokine storm, oftentimes multiple organ involvement, development of acute respiratory distress syndrome, which maybe is at least 30% fatal. So people go on the ventilator and they never come off. Though luckily most cases, 80% or so, are mild or asymptomatic. There's ongoing intense research into the identification of existing agents with acceptable safety profiles for potential use against SARS-CoV-2 for patients with severe SARS-CoV-2 cases. And as the pandemic eases in various countries, there's going to be emphasis now on virus-specific antibody detection. And as I said, vaccine development is ongoing. This is the last section. I just want to go through some of the products that Genetex has invested in, not only to expand and address the SARS-CoV-2 needs of researchers, but also some of our other coronavirus products. I'll go over these quickly. These are you can talk to me directly with specifics, and also all this information is available on our website. We have at least 4 antibodies to detect both SARS-CoV and SARS-CoV-2, and we'll see some examples of that. And these are all against spike. We have 3 antibodies that pick up nucleocapsid, and 1 each against the ORF7A protein and the NSP8. In addition, we're expanding the number of proteins, of recombinant proteins that people use for assay development or for controls and assorted uses in the lab. We also have a large selection of antibodies to ACE2, as well as an antibody against that the cofactor TMPRSS2. And then we also offer camostat mesylate, which was that serine protease inhibitor, which is used to hopefully target TMPRSS2 enzymatic activity. Here are some examples, and the lightning bolt insignia here indicates products that have actually been tested on SARS-CoV-2 infected lysates. And so we have spike antibody showing that, and this antibody was actually originally raised against SARS-CoV, but cross-reacts nicely with SARS-CoV-2, as is the case with this nucleocapsid. Here are some new antibodies, some rabbit polyclonals against spike and nucleocapsid, which work for immunofluorescence. And of course, the ORF7A, again, which binds to recognize both SARS-CoV and SARS-CoV-2. SARS-CoV product catalog, many of the same products, but again, these are antibodies that can be used for research into SARS-CoV. And then we also, in addition, have antibodies to MERS-CoV. Again, the lightning bolt indicating testing on actual virus-infected samples. In terms of Genetex's catalog for cytokine storm, this is a subset of products that in our large immunology catalog. So antibodies, of course, to IL-6. We have antibodies to IL-6 receptor also, IL-1 beta, IL-10, IL-8 GCSF, TNF alpha and beta, interferon gamma, MCP1, and then IP10. Clearly, this is just a subset of what we offer. Genetex has feels that this coronavirus will be an ongoing issue. SARS-CoV-2 certainly, and the concern is that there will be additional coronaviruses that are released. We've had 3 major plagues of coronaviruses within the last 20 years. It's hard to believe this will be the last. So we've invested in continuing to expand our coronavirus line. So specifically for SARS-CoV-2, there'll be new antibodies that are going to be out very soon, this month or the next, or into June. Envelope membrane, NSP1, non-structural protein 1. These are the two key proteases, NSP3 and NSP5. NSP12 is the RNA polymerase. And then ORF8 and ORF10. We have new recombinant proteins, including envelope membrane nucleocapsid. These are in development. Actually, the envelope, I think, just came out. And then spike protein, S1, S2, and then different differentially tagged subdomains of spike, as well as for NSP5, one of the proteases, ACE2, and then something that's relatively new, which might be useful to certain people or certain researchers, would be basically paraffin blocks using HEC293 cells, HEC293 T cell or 293 cells, overexpressing the different structural proteins. And so these are all products that are either already in the catalog or will be soon, hopefully to expedite research and come to some answers. Thank you very much.